Hello students, today's topic for discussion is genome imprinting. This figure gives you idea about imprinting, means how the genes are going to be imprinted into the next generation. Imprinting is nothing but the genes which are expressed in a parent, and they are expressed in a origin specific manner. Either they are from mother or from the father. And obviously there is a marked origin of a parent, as I told you, or they are stamped. Either they are from the mother or from the father, which is during the formation of egg and sperm cell. Then how this stamping is done? This is with the help of methylation. Again, this is a reaction where small molecules, and they are the methyl groups, methylation, that is methyl groups, they attached to the certain segments of DNA. And here, if the genes are partially imprinted when alleles from both parents are differentially expressed rather than complete expression. This genomic imprinting or genome imprinting is demonstrated in fungi, plants as well as the animals, and as far as in mice the 260 imprinted genes are discovered, whereas in human 220 genes are discovered for the imprinting function. How does it works then? So with the help of this genomic imprinting, we can identify the origin of the specific gene, either it is from parental, that is paternal, or maternal. The defects in the genomic imprinting, they are caused by either mutation, deletion, duplication, uniparental disomy, some EP mutations, and they results into the some syndromal imprinting disorders. And this imprinting involves DNA methylation as well as the histone methylation without altering the genetic sequence. And here in this process, the genomic domains are regulated by differentially methylated region, that are the DMRs. And here this figure gives you DNA methylation. So small attachment of methyl groups to the basis of DNA, and it occurs at the cytosine that follows guanine at CpG dinucleotides. So we'll go in detail for the genomic imprinting in angiosperm. As we know, there is a double fertilization in angiosperm. So from the male gamete, so in pollen tube there are two male gametes, and from this one male gamete fuses with the egg cell, and another one is with the diploid nucleus. So during fertilization, the egg cell, or we can say a second separate fertilization event, that gives rise to the endosperm, and one male gamete that fuses with the embryo, or that gives rise to the embryo. So here this second separate fertilization event gives rise to the endosperm, and this nourishes the embryo. Double fertilization it is called because there is two times of fertilization. And this second event of the fertilization, where endosperm is formed, which has a crucial role for providing nutrients and signals for its growth, that is for the viable seed formation and its growth. And this is formed often from the fusion of two maternal cells with the male gamete. And obviously this is triploid in nature, and it forms the triploid genome. So in flowering plant, the central cell is homodiploid and the resulting endosperm is triploid, and that is why it is genetically distinct from the diploid embryo. We know the first male gamete that fuses with the egg cell and that forms the embryo, that is diploid in nature, but endosperm is triploid in nature. So here this ratio of genetic information, as the two components, they are from the maternal parent and one is from the paternal. This ratio is critical for the seed development as well as for the nutrition, and these deviations from the ratio of 2:1 in the endosperm cause endosperm failure in interploidy crosses. What does it mean? And that forms the triploid block effect. So when this endosperm there is a crossing with the lower ploidy level plants, then there is a failure of fertilization or they are not viable. So this increased contribution of maternal as well as the paternal genome, this shows two opposite effect. If there is a contribution or major contribution of a maternal genome, then that inhibit proliferation of endosperm, but when there is a paternal contribution is more, then there is a endosperm is in excess amount or that proliferates the endosperm formation. So how does there is a relation between imprinted gene and triploid block effect? As I told you, these imprinted genes are responsible for a triploid block effect. And what does it exactly do? It prevents hybridization between diploids and autotetraploids. Then what is the triploid block effect? Here, mating with the partners of a lower ploidy level, they cause incompatibility into the endosperm, and what it does, so that leads to the formation of a non-viable progeny, and this is nothing but the reproductive barrier because the progenies are non-viable. But if that progeny they overcome the triploid block effect and they grow as a sporophyte, even they face the problem during the meiosis. This triploid block effect also shows the lack of chromosome pairing that leads to the formation of uneuploid gametes. Sterility is there, unbalanced chromosome sets, so that autoploidy is there. Then this triploid block effect, which is manifested in the endosperm, that constitutes ephemeral tissue particularly sensitive to the ploidy misbalance, and this is why there is no hybridization between diploids as well as the autotetraploids. Then what exactly these imprinted genes do into the specific events? So in evolution of endosperm specific gene, obviously they are, or the genes which are in the endosperm, they are located at a specific form or specific place. So as a location in the vicinity of a repeat or transposon that advance imprinted expression during the seed formation, and they are silencing in the sporophytic organs because of their specificity as they are originated into the endosperm. So they express only during the seed formation and not in sporophytic organs. And because of this gene will lose its functional role during the vegetative life phase, that is sporophytic organs, and because of that it is solely endosperm restricted function. But at the same time these genes regulate the nutrient flow, which has an advantage to control nutrient allocation between the mother and the progeny, which is of prime importance for development of the embryo in case of a human being or the fetus, or in the plants or the angiosperm for the development of the embryo of the seed. Here this figure gives you idea about how this genomic imprinting, that is stamping, which is done with the help of a methylation, and how does it affect. So here in the mammals there is a placenta, okay, that provides the nutrition, and if there is a maternal chromosome or maternal chromosome imprinting is there, they represses the embryo growth or the proliferation of the embryo. And if they are from the paternal parent, the paternal chromosome, they promotes the growth of the fetus by providing the nutrition. And in case of a plant, if the chromosome they are maternal, that is from the mother, so they represses the formation of the embryo. And here if they are from the father or from the paternal chromosome, they promotes the endosperm formation. So this is again how the chromosome, either they are originated from mother or from the father, how they helps for the development of the embryo as well as the endosperm and the seed. This is one theory known as a kinship theory or parental conflict theory, where maternally express the imprinted gene. So whatever the genes imprinted from the mother, they limits the nutrient flow to the embryo. But when the imprinted genes they are expressed from the paternal component or from the father, they promote the embryo growth. That is why this is known as the parental conflict theory. And the first slide that you have observed, that mother and father they are stretching that DNA, and this is inconsistent with the differential effect of maternal and paternal genome into the interploidy crosses. And that's why this theory is of prime importance while we are studying the genomic imprinting. Here one important gene, and that is known as the IGF2. So this gene is specifically imprinted gene and specific only for the paternal component. So they are expressed only from the paternal chromosome. Similarly, the another H19 gene, which is expressed only from the maternal chromosome. So this figure you can observe how this IGF2 gene is switched on and switched off. And the last figure, where again you will get how this imprinting message to the fetus is given either from mother as well as the father. So the father, when this IGF2 gene is switched on, so that helps for the development of the embryo, and he is giving the message that grow big, strong, use all the maternal resources, and the future babies may not have his alleles. And here when this IGF2 is switched off, and the same time another, that is IGF2R, if it is switched on, and it reduces the effect of IGF-2, which is switched on only into the paternal parent. So the mother is saying that grow well, but leave some resources for me and my future babies. So this is all about the imprinting of a genome, or we can say stamping of the genome, where the genes which are imprinted into the next generation, they are marked with the help of a methylation process. Either they are originated from the mother or from the father. So with this I'm going to stop here. We'll meet again with the next video. Thank you.